channel, and in this video we are going to discuss the actual mitosis, in which a nuclei or DNA divides and move to the two daughter cells, or the whole cell divide. So these are characterized by the four different stages. Number one is the prophase, in which the chromosome condensed with the help of histone protein. You must be well familiar with histone protein because we have already discussed this in the previous videos. Then there is a formation of spindle fiber, and it is formed from the centrosome. That is the organelle present in animal cell but not in plant cell. In plant cell, a spindle fiber is formed by the protein present in the cytoplasm. In the metaphase, the chromosome align themselves at the equator, means the spindle fiber is formed, and in the center of the spindle fiber they are going to align themselves. Next one is the anaphase, the sister chromatid separates, means if this is the chromosome, if you have a look at the diagram of chromosomes, these two sister chromatids are going to get separated, so each move to one end, like two different poles. And the last one is the telophase, in which the nuclear membrane reform, the chromosome decondensed into the DNA thread-like structure, means they uncoil themselves. Let's go into the detail of each one of the phase. So the prophase, prometaphase, metaphase, anaphase, telophase are the cytokinesis. Let's go into the prophase. So first of all, the DNA, it condensed to form a chromosome. So it is quite visible when stained. If you want to have a look at the live process, you can easily observe it. Number two, if you look at the structure, there are two identical sister chromatids that is formed by the replication in the S phase. So if we look at this one, so we have a sister chromatid. So each one is called as chromatid. So these two are the sister chromatids. Now they are going to get separate because these are the replicated copies. So they move, each move towards the opposite end. So these are connected at the center here, which is called as centromere. Now the center of centromere is actually, means if this is the centromere, here there is a center, this one is called as kinetochore. Now, kinetochore is actually, you can say, the gluing point where it binds with the spindle fiber. Okay, so what happened in prophase? Now the chromosome condensed, the nuclear envelope is now going to disappear. So this, let's say, nuclear envelope, if they are in nucleus, they are going to be like this one. Your nuclear envelope is going to be disappeared, and you have now the chromosome. I'm drawing only two, you can draw more. So the, how the spindle fiber is formed, that is formed by the centrosomes. They replicate, they replicated in G2. We have discussed this in the previous video as well, and each move toward the other end, means the opposite poles. If this is a cell, so one centromer would be here, so this is a nucleus basically, but let's consider a cell now, and the other one would be here. So the microtubules would be originated from here, here, and here, and then they form like a complete spindle. We'll discuss this later. So a spindle fiber microtubule form from centrosomes, and the nuclear envelope basically break down into vesicles, mean it's going to disappear. Now there are two type of fibers that are going to originate from these centrosomes. There's no need to go into the detail of these two, but keep in mind one is the kinetochore fiber, where the chromosome is going to, sorry, kinetochore fiber. Here the chromosome is going to get attached, and they align themselves in the very center. Okay, Ji. Next is metaphase. Metaphase, the centrosome actually reach the opposite poles, and the spindle fiber, they extend themselves, and there is a formation of a complete spindle. Let's make it, if this is a cell. Sorry about my cell. This is the centrosome. Here we have centrosome. Now a fiber is going to originate and they form a spindle. Now here the chromosomes are going to get attached. So in metaphase they are going to align themselves in the very middle at the kinetochore. So kinetochore is going to be this joining point. So the spindle fiber attached to centromeres and each sister chromatid connect to spindle fiber from opposite poles. Now what happened in anaphase? These spindle fiber they move back into the centrosome, which actually results in the separation of the sister chromatids. Let's talk about the anaphase. So this is characterized by the separation of sister chromatids. So if this is a cell and here we have centrosomes, now here we have the fiber, let's say, and now the sister chromatids are going to get separated. So if they are like aligned like this, now when these fibers move into the opposite direction, they actually break off from each other. Let's do this. So one moves here and one moves here. Similarly here, if we just make it like this, one moves here and one moves here, and then they actually move into the opposite direction as the fiber is contracted towards the centromeres. So the spindle fiber shortens, the microtubule contract, the separate chromatids now pull to the opposite ends. Now if you look at the cell would be like this, that at one end there are the different sister chromatids. So now keep a look on them. If they are like, if you consider them two cells, now each one has two chromosomes. So next one is the telophase. Telophase means they move entirely toward the opposite ends and the chromosome decondense. Now these chromosomes again they recondensed into DNA, not in, let's make two here. Sorry about my coiling, but let's consider this one. Nuclear envelope reforms means that the one disappeared is going to be reformed like this, and now it would have two nuclei, sorry, means one here and one here with the DNA. Let's say it is not condensed. So spinner fiber breakdown. Now the two cells need to be divided. Now how they are going to divide, we are going to discuss that in cytokinesis. So in cytokinesis, it actually there is this division of cytoplasm. So each daughter cell receive one nuclei. So in animal cell, what happens? It's a simple round cell. Let's say it has two nuclei, one at this end, one at this end. Now it start to move from here, pinches actually from here and here, and this is called as cleavage furrow. And from this cleavage furrow they are going to divide into two cells. So a new cell wall form between the nuclei of the, sorry, in animal cell a cytoplasm pinches and the cleavage furrow, while in plant cell vesicles originate, and these vesicles make a cell plate that you can see in the center. Here is a cell plate. This cell plate is now going to separate the two cells. So a new cell will form between nuclei from the vesicles and they get separated. So this is all about the mitotic cycle. If you want to learn this in detail, you can contact at the number given in the start of the video. So see you in the next video. Till then, goodbye.